Welcome back to the deep dive. Today we are attempting one of the greatest visualization challenges in human anatomy.
I think so.
We're taking a deep dive into the inner ear. This is the structure that's hidden deep within the, notoriously dense petrous temporal bone, and it performs the twin essential functions of hearing and balance.
It's not just hidden. It's a masterpiece of biological precision. It's designed to operate at nanoscale sensitivity.
Wow.
When you look at diagrams of the inner ear, you see this impossibly complex, sort of interlinked 3D network of bony canals, ducts, and spiral chambers.
Right, it's so confusing.
It is. So I think our mission today is to try and translate that three-dimensional architecture into a clear mental blueprint for you. We'll be focusing specifically on the 6 specialized mechanosensory patches that convert fluid movement into electrical signals.
6 sensors all working at the same time. I mean, the level of detail here, the bony labyrinths, the fluids, the hair cells, it can feel pretty overwhelming.
Oh, absolutely.
So we're going to act as your guides, really, to help you navigate this tiny, tiny world.
At its core, the inner ear houses the labyrinth. And, well, that term really does perfectly describe its structure.
A maze.
It's a maze. This system is a network of interconnected bony cavities, and inside that is another network of delicate membranous sacs and ducts. This membranous part is filled with fluid, and it contains all the sensory structures that govern our hearing and balance.
Okay, let's unpack this then. So we'll navigate the dense protective bony framework first.
Yep.
Then we'll tackle the specialized fluid system that literally, powers the entire process. And finally, we'll explore how those delicate hair cells convert the slightest vibration into the electrical signals that race up the vestibulocochlear nerve.
Sounds like a plan. So the foundation is the osseous, or bony labyrinth. This structure is actually denser and harder than the bone around it, the petrous temporal bone, which gives it just phenomenal protection.
So think of it as the ultimate fortress. And this fortress has, 3 main cavities that shape everything inside?
That's right. The vestibule, the 3 semicircular canals, and the coiled cochleae.
Okay.
And inside that bony shell sits the membranous labyrinth, a series of continuous interconnected sacs and ducts.
And the space between the rigid bony wall and that flexible membranous part, that's what's so important.
Absolutely critical, because that's what establishes the unique fluid dynamics of this whole system.
And this is where it gets really interesting for me. The 2 fluids, they are kept strictly separate, and they are chemically opposite.
Completely opposite.
So first, there's perilymph. This fills the space between the bony wall and the membranous sac.
Right.
And its ionic composition is like standard extracellular fluid, so low in potassium, high in sodium.
Now contrast that with endolymph. This is the fluid that fills the inside of the membranous labyrinth. Crucially, its ionic makeup is like the fluid inside most of our cells, cytosol.
So high in potassium.
Exactly. High in potassium, low in sodium. And this separation is not just some chemical curiosity. It is the absolute power source of our hearing.
You keep calling it the power source. Why is this difference in potassium concentration so important? How does it translate into power?
Because it maintains this staggering endolymphatic potential. Due to that high potassium in the endolymph, it registers about 80 millivolts more positive than the perilymph.
80 millivolts, okay.
So when a hair cell sits between these 2 fluids, its top, its apex, bathed in endolymph, its base in perilymph, you create a total electrical driving potential of up to 150 millivolts across that tiny hair cell apex.
150 millivolts. Is that the largest potential difference across any cell membrane in the body?
It is certainly among the largest sustained potentials. And you need that extreme voltage because the hair cells have to detect the most minute molecular vibrations from sound or head movement.
Ah, I see.
That 150 millivolts ensures that when the little mechanical gates on the cells open, the rush of positive potassium ions is immediate and massive. It just maximizes the cell sensitivity. It's an evolutionary necessity for acute hearing.
That context makes the anatomy so much clearer. So now that we've got the chemical power source, let's see how that energy is applied, starting with the balance center, the vestibule.
The vestibule is the central chamber. You can picture it as this sort of flattened ovoid room.
The traffic hub.
The traffic hub of the inner ear, exactly. It's situated just medial to your middle ear cavity, and it connects the cochlea in the front to the semicircular canals in the back.
Okay, so on the lateral wall of this hub, we find the oval window, right? That's where the stapes bone fits in to transmit the vibrations.
That's the one. And on the other side, the medial wall, we find 2 specific little recesses that house the main sensory sacs for linear balance. You've got the spherical recess anteriorly, which contains the saccule.
And the elliptical recess behind that contains the utricle.
Precisely. And moving backward from the vestibule, we find the 5 openings for the semicircular canals.
And there are 3 of them, as the name suggests.
3 of them anterior, posterior, and lateral, each one forming about 2/3 of a circle. The anterior and posterior canals are vertical, the lateral ones horizontal, but they all share a crucial feature, a terminal swelling known as an ampulla. That's where the rotational sensors are housed.
And if you're trying to picture the plumbing here, you should know that the anterior and posterior canals actually join up before they re-enter the vestibule.
Right, yeah.
They form this single tube about 4 millimeters long called the crus commune, the common limb. It's a really efficient piece of biological architecture.
It really is. Now, for function, the vestibular system handles 2 types of balance information. First, static and linear balance, say your sense of gravity and straight line acceleration.
Okay.
This is handled by the sensory patches, called maculae, that are inside the utricle and saccule.
So let's visualize how that works. The utricular macula, that one's larger and lies mostly horizontally flat.
That's right.
So because of that flat orientation, it's the main detector for linear acceleration in the horizontal plane, like slamming on the brakes in a car or tilting your head to the side.
Exactly. The saccular macula, on the other hand, lies mostly in a vertical plane. So it is highly sensitive to linear acceleration in the vertical plane.
Like in an elevator.
A fast elevator, or jumping up, yes. That specific sensation is largely the saccule reacting. It's a major gravitational sensor.
So if you're just standing straight up, the saccule is highly active, basically telling your brain where down is.
Precisely. And the mechanism for both is the same. The maculae are covered by this dense, gelatinous structure called the otolithic membrane.
And this membrane is embedded with thousands of tiny calcium carbonate crystals, the otoconia.
The ear rocks. And when your head moves, the sheer inertia, the mass of this crystalline layer, causes it to lag behind the surrounding fluid.
And that lag mechanically deflects the underlying hair cells and fires off a signal.
You've got it.
Okay, that explains steady motion. But what about spinning? We need the third dimension, dynamic balance, detecting rotation.
That's the job of the cristae, these sensory crests, located inside the ampullae of the semicircular ducts. The fluid dynamics here are just fascinating. When you start turning your head, the walls of the duct move, but the endolymph inside briefly lags behind because of inertia.
And that relative flow pushes on something.
It pushes on a gelatinous structure that covers the crista, which then stimulates the hair cells.
What's truly brilliant is the functional coding. The 3 canals are oriented roughly at right angles to each other.
Like X, Y, and Z axes.
Exactly. This geometry ensures the brain gets input about rotation in absolutely any direction.
And they work in functional pairs to maximize sensitivity. So, for example, if you quickly turn your head to the left,
Okay.
the endolymph flow excites the receptors in the left lateral canal. But at the same time, it inhibits the complementary receptors in the right lateral canal.
So one side fires up, the other powers down.
Right, and this opposing signaling gives the brain this powerful, unambiguous signal about the direction and speed of the turn.
Incredible design. Okay, now, let's follow the labyrinth forward from the vestibule into the most famous structure, the cochlea, named after the Greek word for snail, koklos.
And it looks just like one. It spirals about 2 3/4 turns around a central bony core, the modiolus.
Right.
And this bony spiral lamina projects out from the modiolus, but it only partially divides the cochlear canal. This partial division is critical
Why is that?
because it creates 3 fluid-filled longitudinal channels that run the entire length of the spiral.
Okay, so the highest channel is the scala vestibuli, which starts at the oval window.
Correct.
The lowest channel is the scala tympani, which ends at the round window.
And sandwiched right in the middle, running between those 2 perilymph-filled channels, is the cochlear duct, or scala media.
Ah, and that's the one that contains that special high potassium endolymph, the power source for hearing.
That's the one.
So keeping those fluids separate requires 2 very specific membranes. You have Reisner's membrane, which is gossamer thin, separating the scala media from the scala vestibuli above it.
And then below it, forming the floor of the scala media, is the vital basilar membrane. This membrane is, well, it's not uniform at all, and this is essential for telling frequencies apart.
How so?
It is wider and more flexible at the apex of the cochlea, which makes it responsive to low-frequency sounds.
Like a bass drum.
Exactly. And conversely, it's narrower and stiffer at the base, where it responds to high frequencies.
So the basilar membrane is literally an internal frequency map, a biological piano keyboard where different sound pitches peak at different locations along the spiral.
That's a perfect analogy. And sitting directly on top of that basilar membrane is the sensory epithelium itself, the organ of Corti.
And this sensory structure is so meticulously organized, it has a single delicate row of inner hair cells.
The true messengers.
They provide the vast majority of auditory signals sent to the brain. And then there are 3 or 4 supporting rows of outer hair cells.
And all these cells are structurally supported by pillar cells that lean against each other to form this triangular tunnel of Corti. The tight junctions between the tops of these cells form a strong seal, the reticular lamina.
Which is what keeps the fluid separate at the business end.
It ensures the tips of the stereocilia are bathed in that high-voltage endolymph, while the cell bodies are bathed in perilymph.
Okay, so how does the hair cell actually convert that fluid vibration into a nerve signal? Let's get into the mechanotransduction.
Right. The top surface of the hair cell has up to 100 of these modified microvilli called stereocilia. They're arranged in a neat little staircase.
And they're all physically connected by these incredibly fine filaments called tip links,
The tip link is essentially the mechanical tripwire.
which opens the gate.
Precisely. Fluid movement from sound waves deflects the hair bundle towards the tallest stereocilium. This puts tension on the tip link, and it mechanically yanks open the TMC1 transduction channels.
And then that 150 millivolt potential.
It drives an immediate influx of potassium and some calcium from the endolymph. The cell depolarizes and releases glutamate onto the afferent nerve fibers, and you have sound.
That explains detection, but it doesn't quite explain the incredible sensitivity we have. We mentioned the outer hair cells. They are the engine of the cochlear amplifier. How do they work as a motor?
The outer hair cells, or OHCs, are unique. They contain a protein called prestin.
Preston.
Prestin allows these cells to change their length almost instantaneously. They contract and expand electrically. It's a form of piezoelectricity at frequencies up to thousands of times per second.
So they're physically moving.
They are physically moving. The OHCs act as a mechanical motor, actively pushing and pulling on the basilar membrane, dramatically boosting the vibration signal for the inner hair cells.
So the inner hair cells are the passive microphones sending the signal, but the outer hair cells are the active amplifiers. They fine-tune the mechanics of the whole system.
They do. They increase sensitivity and frequency discrimination up to 1,000-fold. Without functional OHCs, hearing is severely impaired.
Wow.
So moving to the communication wires. Innervation is handled by the 8th cranial nerve, the vestibulocochlear nerve.
The auditory part, the cochlear nerve, originates from bipolar neurons in the spiral ganglion, which is tucked away inside the modiolus.
In a little structure called Rosenthal's canal. The vestibular part comes from the vestibular ganglion, or Scarpa's ganglion, and that supplies the maculae and cristae.
But there are also wires coming back from the brain, right?
Yes, the efferent fibers, part of the olivocochlear system. This is the brain's way of turning the volume down, modulating the response of the hair cells.
How does he say it?
The medial efferents act directly on the outer hair cells, using acetylcholine as the neurotransmitter, essentially telling them to amplify less.
The power supply for this whole critical area is quite singular, isn't it? It's just the one labyrinthine artery.
Pretty much. It usually branches off the basilar artery, or AICA. And that single source really highlights its vulnerability. The maintenance of that 150 millivolt potential depends completely on a robust, continuous blood and oxygen supply from this one artery.
Makes sense. Okay, since the system is so precise, it must be highly prone to specific mechanical problems. Let's touch on a couple of clinical connections, starting with the most common cause of vertigo, BPPV.
Benign Paroxysmal Positional Vertigo. This is a classic mechanical failure involving those otoconia we talked about.
Key ear rocks.
The ear rocks. For various reasons, they can get dislodged from the utricle's macula and fall into one of the semicircular ducts, most often the posterior one, just due to gravity.
And these rogue crystals just float around in there.
Right, they're called canales. And they inappropriately stimulate the rotational sensor in that canal's ampulla, causing intense but short vertigo when the head moves into certain positions.
A nightmare. But the treatment, the Ebley maneuver, is entirely non-surgical. How does that work against the mechanical problem?
It's purely leveraging gravity and anatomy. The patient is moved through a very specific series of head and body positions. These are calculated to use the semicircular canal's own shape to sequentially roll those loose crystals out of the sensitive duct and back into the vestibule, where they can be reabsorbed.
Just rolling them back out. Incredible. We also see sensorineural hearing loss, which is actual damage to the sensory hair cells themselves.
Yes, from noise trauma, certain drugs, or genetics. And actually, a significant number of genetic hearing loss cases involve mutations in a protein called connexin 26.
So it's not always the hair cell's fault.
Exactly. This highlights that deafness can be a failure of the system's power maintenance. Connexins form gap junctions needed to recirculate potassium ions back to the endolymph to maintain that critical 150 millivolt potential. If that pathway breaks, the hair cells basically starve.
And for profound hearing loss, where hair cells are completely gone, the most advanced solution is the cochlear implant.
Correct. The CI is remarkable because it skips the natural system entirely. An electrode array is surgically inserted into the skull tympani. It bypasses the damaged hair cells and directly stimulates the surviving spiral ganglion neurons.
So it's electrically replicating the frequency map along the cochlea.
That's exactly what it's doing.
What an extraordinary journey through this hidden world. So to quickly recap, we navigated the dense bony labyrinth. We discovered how the contrasting fluids, perilymph and endolymph, generate that immense electrical charge.
150 millivolts.
And we explored the dual nature of balance, the utricle and saccule maculae for linear motion, and the cristae in the semicircular canals for sensing rotation.
And finally, we saw the highly tuned cochlea, where the basilar membrane acts as a frequency filter, and the outer hair cells use that motor protein, prestin, to act as active amplifiers, giving us our acute sensitive hearing. This does raise an important question, though.
Given all this intricate biological engineering, what is the future frontier?
Well, given the incredible precision and speed of the outer hair cells, how they change shape thousands of times per second, how might future therapies move beyond bypass technology, like the implant, and instead leverage the body's own electromechanical properties to fully regenerate or restore that function?
So not just bypassing the problem, but actually fixing it at the source.
Exactly. That level of biological fidelity, that's the ultimate challenge.
A fascinating and provocative thought to end on. Thank you for joining us on this deep dive into the anatomical basis of hearing and balance. Keep learning, and we'll see you next time.